Hi everyone, this is Jonathan Cedillo, student at Grand Canyon University, and today I'm presenting to you vasopressin role in urination behaviors of male mice. Presentation for the class Biology 520 Concepts of Human Physiology 2, Professor Dr. Joan Tassoni. In the following article, contrasting central and systemic effects of arginine-vasopressin on urinary marking behavior as a social sign in male mice, the scholars Higuchi and Arakawa claim that arginine-vasopressin, or AVP, is an important molecule located at the neurohypophysis that influences animal behaviors. AVP plays a critical role in the modulation of important signaling processes, for instance, urinary excretion to mark territory dominance with their scent. For the neurohypophysis, AVP is delivered to the circulatory system. Once there, it can show antidiuretic hormone activity. The kinetics of AVP suggests that the main objective of this hormone towards marking territory is not linked to the activity of AVP in the regulation of an osmotic environment. Using mice as a target for experimentation, peripheral administration of AVP downregulated urinary activity, including marking with their scent. On the other hand, central infusion of AVP assisted the progress of marking and no signs of modification on urination. However, the antagonist molecule for the AVP1A receptor constrained the marking. Administration of AVP injection to mice resulted in secondary effects such as sneezing and itchiness. These secondary effects provoked the uninterest from scent marking of mice. Mice that were stimulated during post-testing showed results of high levels of AVP synthesis at the midbrain. However, that was not the case in the circulatory system. Central AVP controls scent marking together with other behaviors. This is suggesting that AVP is released to the circulatory system, causing activation of neural connections, to later the peripheral AVP acting as an antidiuretic hormone for urinary scent marking. In mice, the male accumulates proteins in the urine for urinary marks. These behaviors are utilized by these animals as a form of territorial dominance and social interaction, hence the importance of the behavior. Specialized glands in the male mouse create pheromones that are in the urine that later will be released at any location to claim territory. This action warns others from the same species the presence of a potential opponent. Arginine-Vasopressin, or the already mentioned AVP, is a primitive neuropeptide that is found across different species of the world. Along the periphery, AVP activity regulates cardiovascular osmosis together with antidiuretic and vasoconstriction of capillaries activity. In addition, scholars claim that AVP acts as a neuromodulator in sociosexual conducts. As already stated, scholars claim that AVP has a crucial role in the control of urinary activity together with marking territory, which is not linked to the activity of peripheral AVP in the containment regulation of urine waste. When mice sense the need to mark a region of territory, arginine-vasopressin is synthesized at the brain and activates scent marking conducts. While AVP is synthesized, this does not produce conductivity of the same towards the circulatory system, which means that peripheral AVP is not stimulated. To analyze the kinetics of AVP process regulation during marking conducts, experts used male mice. These subjects were administered with AVP at the central and peripheral regions with an injection. Then mice was released to initiate marking conducts at the presence of other males as well as isolated. For this research, a special strain of male mice was used, specifically B6 mice, that accordingly to the research, the animals as targets are an inbred strain that possess identical social olfactory cues within the same strain and cannot distinguish subjects of the same strain. Here, figure 1 represents the process of experimentation phase of scent marking and how it was approached. Each mouse underwent 3 trials. In the test, the mice were administered with vehicle, AVP, or vasopressin 1A receptor antagonist, also known as B1AA. Then, mice were introduced to social interaction of a male B6 mouse. Also, mice were isolated after injection. The mice were conducted through three tests under six circumstances, which are drug treatment encompassing vehicle, AVP, or vasopressin 1A receptor antagonist, which is V1AA, and confrontation, anti or social cages, as shown in Figure 1. Only prior experimentation, mice were administered with an intraperitoneal or IP injection of the drug agents 30 minutes before analysis of behavior. The mice were placed at the chambers without any order in specific, isolated or in company of the B6 mice. During experimentation, authors of the article claimed that all mice, including the stimulated ones, were isolated from the group to be placed at the experimental containers 30 minutes before test. The experimental spots were analyzed using 40-watt lights as shown in Figure 2. After mice were injected with the drug of interest, they were placed alone or in company under infrared light during 20 minutes for them to start marking behaviors. Here, Figure 2 shows the results after mice BALB, to be specific, were placed at the chamber under the light and they proceed to mark. After 20 minutes, the paper substrate in the cage was dyed with a 9-hydrogen spray. Then the marking spots were studied. Here in Figure 3, authors of the article explain that after applying an IP injection of the drug's vehicle, AVP or V1AA, 30 minutes before the test, some BALB mice were placed in the testing cage completely isolated or together with a stimulus such as social company of mice B6. In Figure 3, graph A represents the distance traveled by the mice. Graph B represents the time the BALB mice remain at different spots in the cage. Graph C shows the actual scent marks across the substrate paper. Graph D shows us the exocrine mark index. By analyzing this data, scientists observed that the kinetics of BALB mice were higher than when socially stimulated by a B6 mouse than when isolated. BALB with AVP administration has less motion than mice with B1AA. Graph B showed that stillness of subjects demonstrated behaviors that are alike to the mice that had more motion. This is suggesting that the drug has effect over the mice. However, that was not the case in the socially stimulated animals. This means that BALB mice with AVP in the system demonstrated more motionless behaviors in contrast to the mice that were mere injected with B1AA or saline solution. Male mice had a greater marking activity when put together with a B6 mouse than isolated. AVP administration on the periphery demonstrated effects without significant conducts between stimulation and treatment. After analyzing data with a post-hoc test, results showed that mice with AVP had less marking activity when isolated in response to social interaction, in comparison to those mice that were injected with the vehicle. However, the mice that were administered with V1AA antagonists demonstrated increase in scent marking as presented in Figure 3C. Here, in Figure 4, authors of the article explain that urinary scent marking behavior of BALB mice that were intracerebroventricularly or ICB added with the drugs vehicle AVP or V1AA 15 minutes prior testing. These BALB mice were placed at the testing cage isolated or with a social stimulus of a B6 mouse. Figure 4 graph A represents the displacement. Graph B is the time that VALB subjects remain motionless. Graph C presents the numbers of urinary scent marks. And graph D represents the urination that was counted as an exocrine marks index. In figures 4A and B, the motion activity was analyzed at the distance traveled and time of motionless. Mice that were administered with drugs ICB and AVP presented a larger range of movement in both cases scenarios, socially stimulated and isolated as well. In contrast to the subjects which were injected with vehicle treatment, the data was provided by the two-way ANOVA test. Other studies showed that subjects under the influence of AVP administration had a greater activity in kinetics in contrast to the ones that received vehicle drug. However, that was not the case for the subjects with V1AA treatment. In addition, all mice presented alike ranges of motionless times when receiving vehicle, AVP, or V1AA treatments with or without social stimulation. More analysis showed that mice injected with vehicle and AVP created more marking spots when placed together with a V6 mouse than the subjects that were isolated in a cage, while mice under AVP influence presented alike ranges of scent marks when exposed to another V6 mouse or alone. BALB subjects with AVP administration showed more marked activity than mice with vehicle administration when placed in an isolated cage. However, the opposite occurred when socially confronted with a V6 mouse. Further, BALB subjects with a VAA administration presented less marking activity than mice with vehicle treatment when isolated and when socially paired with another mouse. Moreover, a central injection of AVP did not affect the urinary wastes of VALB mice as presented in Figure 4D. This suggested that VALB subjects with AVP or B1AA administration loaded fewer marking spots during testing compared to the mice who received vehicle administration. BALB subjects who got AVP administration delivered less wastes along the testing than those with V1AA injection. Here, in Figure 5, conducts of BALB mice that received an intracerebral ventricularly ICV administration with either vehicle, AVP, or B1AA 50 minutes before the test are shown at the testing cages, either socially stimulated or isolated. Graph A presents the time spent in poking snout in a wire mesh partition. Graph B shows the times BALB mice presented high cough or sneezing activity. Graph C is the time they spent scratching, and Graph D presents the grooming during testing which was conducted manually. Further, the time for nose poking showed an upregulation of the activity when socially stimulated in contrast to the isolated subjects. Mice that were injected with AVP upregulated nose poking in contrast to the mice that were administered with vehicle treatment. Particularly, BALB mice with AVP injection showed a chronic hiccough and sneezing behaviors as presented in Figure 5V. When socially stimulated than isolated, this type of activity was greater than vehicle and V1AA treatments. In addition, VALV subjects with AVP injection showed chronic scratching behaviors as presented in Figure 5C. During social interaction, the mice did not present a difference in the time spent for scratching, in contrast to the subjects that received vehicle treatment and V1AA. Further, there was not a significant difference between the groups in the time spent on grooming as presented in Graph 5D. Authors of the article explain that a long testing time, AVP is a critical regulator of urinary excretions. Thus, central AVP expression is carried at central locations of the gray matter and delivers AVP into the blood. Scholars determined that the levels of expressed AVP in the hemispherical midbrain regions and circulatory system prior subjects were placed in the non-testing chambers, and when in the testing chambers isolated, or when socially stimulated with a V6 mouse. Here in Figure 6, the concentration of AVP in plasma and midbrain of VALB subjects in non-testing chamber and testing cage isolated or socially stimulated is presented. Figure 6, Graph A presents the distance travel of the subjects. Graph E is the scent marking spots after 20 minutes of displacement. Graph C shows the AVP in the plasma. Graph D shows the brain tissues containing the bed nucleus of the stria terminalis. Lateral septum, the posterior striatum, anterior and posterior hypothalamus, thalamus, and amygdala complex. Graph E is the interrelationship of plasma AVP concentration of the number of sense marking spots. Finally, Graph A represents the interrelationship of brain AVP concentration and the number of sense marking spots. Conclusion. According to the article Contrasting Central and Systemic Effects of Arginine-Vasopressin on Urinary Marking Behaviors as a Social Signal in Male Mice, Arginine-Vasopressin, or AVP, is a peptide that exists in the neurohypophysis. This peptide can regulate the critical conducts of many animals. Interestingly, AVP controls social behaviors in male mice, for instance, urinary secretion together with their marking behaviors. Along the periphery, AVP is delivered to the portal bloodstream and acts as an antidiuretic hormone. The kinetics of the peptide have a significant role in the AVP stimulatory activity of urinary marking, which is separated from the role of AVP in the accumulation of osmotic conditions. Here, in this study, by using BALB mice as the subjects, the peripheral injection downregulated urinary and marking activities. However, a central administration of AVP upregulated urinary marking. Nonetheless, it did not have a significant effect over urination. On the other hand, the antagonist of AVP1A receptor inhibited marking behaviors. In addition, central AVP injection in these subjects showed normal conducts of sneezing and scratching, especially when it's socially stimulated with the presence of a B6 mouse in the same chamber. Marking resulted affected by the social interactions together with the sneezing and the scratching reactions. Studies also demonstrated that AVP expression increased at the midbrain region, however, not in the circulatory system in post-test time, especially when socially stimulated. Scholars of this article explain that central AVP controls urinary marking and other conducts in a dose and situation-dependent manner. Finally, scholars state that the sequential process implies that centrally synthesized AVP might be secreted into the circulation following immediate neural processes, and the peripheral AVP acts as an antidiuretic hormone on urinary marking behavior. Thank you for watching my presentation. See you next time.